hope you're having a lovely day. Thank you for tuning in to another video. Today we're going to look at how your body communicates within itself. Don't forget to check out our next video on how drugs, including medication and alcohol, interfere with these communications and can lead to addictions. Firstly, we will look at nerve impulse and how these allow communications across your body in a chain reaction before looking specifically at synaptic transmission on a granular level. So let's start with the nerve impulse. Neurons communicate through electrochemical signaling. There's electrocommunication within neurons and chemical between neurons. A nerve impulse is the electrical message that is transmitted down the axon of a neuron, the impulse that travels within nerve cells. This transport is quick and without loss of energy. It is regenerated along the axon. Electrical currents in the body reflect the flow of ions. The nerve impulse can be broken down into three sections. Firstly, resting potential. Secondly, action potential. And thirdly, communication between neurons. So let's jump into resting potential. What is resting potential? The resting potential of a neuron refers to the state of the neuron prior to the sending of a nerve impulse. When the neuron is at rest, the membrane maintains a specific electrical gradient. This is the difference in the electrical charge inside and outside of the cell, and this is approximately negative 70 MV. The neuron maintains the negative 70 MV charge due to the membrane being selectively permeable, which means that it only allows some chemicals to pass more freely than others through the membrane. Water, oxygen, and carbon dioxide can pass freely. However, sodium, potassium, and chloride pass through channels which are protein molecules in the membrane. When the membrane is at rest, sodium channels are closed. Potassium channels are partially closed, which allows the slow passage of potassium, and chloride channels are partially open. The electrical gradient is maintained by active transport, which does require energy. The sodium-potassium pump continually pumps 3 sodium ions out of the cell while drawing 2 potassium ions into the cell, and sodium ions are 10 times more concentrated outside than inside the neuron, whilst potassium ions are more concentrated inside than out. Passive transport, otherwise known as leakage, happens when sodium channels are partially open. When at rest, 2 forces try to push sodium ions into the neuron. The electrical gradient pulls positively charged sodium ions back into the negatively charged neuron through the concentration gradient. A greater number of sodium ions outside means that there is an increased probability of sodium moving in rather than out, but the channels are currently closed during resting potential. These 2 forces instead compete with each other for potassium ions. The electrical gradient pulls positively charged potassium into the negatively charged neuron. Through the concentration gradient, there is a greater number of potassium ions inside, which means there is a greater probability of potassiums moving out rather than in. Now let's look at what happens when these channels open. An action potential can be created. So the resting potential remains stable until the neuron is stimulated by either hyperpolarization, which is when the membrane potential becomes more negative, for example, to negative 80 MV or negative 90 MV. Or depolarization, which is when the membrane potential becomes less negative, for example, negative 60 MV or negative 55 MV. The threshold of excitement is negative 55 MV, and this refers to the level above which any stimulation produces a massive depolarization and the action potential is produced. Sodium channels are voltage-activated channels. This means that the membrane channels have a permeability that depends upon the voltage difference across the membrane. When the membrane potential reaches the negative 55 MV threshold, the membrane opens its voltage-gated sodium channels. When sodium channels are open, positively charged sodium ions rush into the neuron and a subsequent nerve impulse occurs from a reduction in the polarization. This means the inside of the neuron becomes positive. At a certain threshold, which is approximately positive 30 MV, sodium channels are quickly closed to prevent even more sodium from rushing into the neuron. The neuron is returned to its resting state by the opening of potassium channels. There is an outflow of potassium due to the concentration gradient of an extremely positive charge inside the neuron. Therefore, the potassium leave and take with them their positive charge. This is hyperpolarization. The sodium-potassium pump later restores the original distribution of ions through taking 3 sodium out and taking in 2 potassium. Sodium ions cross during the peak of the action potential. Potassium ions cross later in the opposite direction, returning the membrane to its original polarization. There is an all-or-nothing law, which states that the amplitude and velocity of an action potential are independent of the intensity of the stimulus that initiated it, which means action potentials are equal in intensity and speed within a given neuron. Stronger or sustained stimulation will not change the size of the action potentials, but will result in a greater number of them. The refractory period is a period of time the neuron resists the production of another action potential. There is the absolute refractory period, which is the first part of the period in which the membrane cannot produce an action potential. This is approximately 1 millisecond. Then there is the relative refractory period, which is the second part in which it may take a stronger than usual stimulus to trigger an action potential. This is approximately 2 to 4 milliseconds. But these are both very important refractory periods as these stop the signal from travelling backwards. For example, if the third neuron experienced an action potential, what would stop it from travelling backwards to the second or even the first one that had started the action potential sequence? And that is why the refractory period is very important at stopping these signals from travelling backwards. As the action potential travels down the neuron, this is called a wave of depolarization. As the action potential travels down the neuron, a wave of depolarization passes to adjacent areas on the membrane. When this reaches the threshold, sodium channels open and the action potential is regenerated. The action potential moves along the axon in one direction because of the refractory period. The movement is not always continuous. At each node of Ranvier, the action potential is regenerated by a chain of positively charged ions pushed along by the previous segment. There are different types of conduction, for example, excitatory conduction. Myelination will also help move the message along quicker. Let's now look at communication between neurons. There is a presynaptic neuron. This is the neuron that delivers the synaptic transmission and is found at the end of the axon. There is postsynaptic neuron, which is the neuron that receives the message, and this is found on dendrites and cell bodies. The synaptic gap has chemicals released from the presynaptic and absorbed by the postsynaptic. Charles Sherrington studied temporal and spatial summation on reflexes in dogs. He was the first person to discover the transmission is chemical. Charles found that the speed of conduction took longer within multiple neurons than within a single neuron. The delay must occur because of communication between the neurons. This gap slows down the communication between them. Otto Loewy stimulated the vagus nerves that control heart rate and managed to slow them down. They transferred fluid from the heart of one frog and added this fluid to a second frog's heart. The second frog's heart also slowed, even though the vagus nerves were not directly stimulated. This shows that nerves communicate by releasing chemicals. During synaptic transmission, presynaptic neurons release chemicals or neurotransmitters which activate the postsynaptic neuron. Each neuron generally synthesizes and releases at least one type of neurotransmitter. Here are some common neurotransmitters and what their relevant roles are within the body. So, acetylcholine is a neurotransmitter used by the spinal cord neurons to control muscles and by many neurons in the brain to regulate memory. In most instances, acetylcholine is excitatory. Dopamine, the neurotransmitter that produces feelings of pleasure when released by the brain reward system. Dopamine has multiple functions depending on where in the brain it acts, and it's usually inhibitory. GABA, or GABA-aminobutyric acid, the major inhibitory neurotransmitter in the brain. Glutamate, the most common excitatory neurotransmitter in the brain. Glycine, a neurotransmitter used mainly by neurons in the spinal cord. It probably always acts as an inhibitory neurotransmitter. Norepinephrine, Norepinephrine acts as a neurotransmitter and a hormone. In the peripheral nervous system, it is part of the fight or flight response. In the brain, it acts as a neurotransmitter regulating normal brain processes. Norepinephrine is usually excitatory, but is inhibitory in a few brain areas. And serotonin, a neurotransmitter involved in many functions, including mood, appetite, and sensory perception. In the spinal cord, serotonin is inhibitory in pain pathways. So let's now look at a more granular level of synaptic transmission. So what happens during synaptic transmission? Firstly, there is the synthesis of neurotransmitters in the cell body and axon terminals from molecules derived from food. For example, acetylcholine provides learning from choline, which is abundant in cauliflower and milk, and serotonin can lead to an uplifted mood, and this is from tryptophan, but is also in milk. Release and diffusion. When an action potential arrives at the presynaptic terminal, calcium ions enter, which triggers the release of neurotransmitters. Neurotransmitters are contained within vesicles. The entering of calcium causes the vesicles to fuse with the membrane and release the neurotransmitters into the synaptic gap. Activation of the postsynaptic receptors. Neurotransmitters attach themselves to a postsynaptic receptor like a lock key system. The combination neurotransmitter and receptor and the location in the brain determine the effects. Inactivation and reuptake. The neurotransmitters either leave the receptor, for example, serotonin can leave through diffusion or reuptake into the presynaptic neuron, or they're broken down by enzymes. For example, acetylcholine is broken down through enzymatic degradation. Importantly, it's worth noting here that epilepsy can be caused because the neurons keep being stimulated as the neurotransmitters are not being removed or broken down. And lastly, synaptic transmission. Postsynaptic activation will open or close ion gates. Slightly open sodium gates leads to depolarization through excitation. Excitatory postsynaptic potentials or slightly open chloride gates leading to hyperpolarization or inhibition through inhibitory postsynaptic potentials. One excitatory postsynaptic potential will not create an action potential, as there must be temporal summation, spatial summation, and excitatory postsynaptic potentials must be greater than inhibitory postsynaptic potentials for an action potential to be created. I hope you guys have enjoyed this video where we've broken down nerve impulses and also looked at synaptic transmissions. Don't forget to check out our next video, which will look at how different drugs might interfere with these communications, such as medication or alcohol. See you guys then. Thanks. Bye.